Hello everyone, welcome back to MedMiracle. Today we are going to discuss about the important topics in biochemistry. First, let's discuss about the reference book. Harper, it's a standard textbook. When I say standard textbook, you have to take it in that sense only. It's a reference textbook whenever you have any controversy or doubt. Otherwise, you don't have to check it out. Many people will say they haven't read Harper in their UG life. Do they have to read from the starting onwards? No, nothing like that. Whenever you have a controversy topic or doubt, just go back and read that topic from Harper. Whatever is that answer mentioned in Harper is your answer. No matter whatever other guidebooks say, whatever it is, the answer mentioned in Harper is your answer for your exam purpose. The next two books, First Aid USMLE and Kepler, both are USMLE textbooks, but the good thing about these books is biochemistry is given really beautifully. In First Aid, it's hardly 20 to 30 pages. Beautiful charts, tables are available, and it's really good to revise topics from that. And if you're asking for a guidebook, I will recommend Rebecca James. And the thumb rule for any guidebook is that you don't have to read A to Z. You have to be very specific. You first go to the previous year questions and then see what all topics are being mentioned, then go back and read those topics. That's it. You don't have to read A to Z. You will feel lost and you will stay lost forever. So be specific before you read the theory topic in any guidebook. Be sure what the questions are, where the questions are coming, and what you have to read from that. So this is one of the charts available in first year, just to give you a summary or just an introduction to the first aid of USMLE. There are a lot of charts and tables available in first aid which will help you in revision. So please go through it. In biochem, the topics you have to stress on first one is cycles, various metabolic pathways then DNA, then you have vitamins and other minerals, and general topics. At least 80% of the questions come from cycles. They ask a lot of questions and a lot of cycles are available. So this is a list of it. We have to go individually and let's discuss what all is important in each of the topic. First, coming to glucose metabolism. In this, there are multiple pathways available. You have to be thorough with all these 5 cycles. Being thorough with these 5 cycles doesn't mean that you have to memorize each and every step and the enzyme involved. No, absolutely no. You have to be very specific. One thing you must always remember is the sites. At what location the so and so pathway is happening. For example, glycolysis is happening in the cytoplasm, TCA cycle is happening in the mitochondria. You have to know the site. That is very, very important with respect to any cycles, not just glucose metabolism. To all biochemistry pathways, you have to know the site. Then coming to ATP production. ATP production is important for glycolysis and for beta oxidation of fatty acid. These two, you have to be thorough with the ATP production. And in Beta oxidation, they mostly ask calculations, like in palmitic acid how many ATPs are formed, in arachidonic acid how many ATPs will be formed in beta oxidation. That's the type of question mostly that come from beta oxidation. With regard to glycolysis, mostly you have to know at which step ATP is being produced. So that is an important point you have to stress on. Okay, so coming to the next topic, irreversible steps. In glycolysis, there is 3 irreversible steps, and it is important because in case of gluconeogenesis, it's these 3 steps which is blocking the formation of glucose. So the body finds out an alternative pathway to bypass these irreversible steps. So the difference between glycolysis and gluconeogenesis comes at these 3 steps, and there is no wonder these 3 steps are frequently asked. Sometimes a question may come like, which of the following is not part of gluconeogenesis, and one of these 3 steps which is part of glycolysis will be asked. So, and vice versa can also happen. So we talked about sites, irreversible steps, ATP production. Now coming to the next point we have to stress is the regulatory steps. Obviously all these metabolic pathways are under the control of hormones, and in case of glucose, insulin and glucagon are the chief hormones which control these metabolic pathways. So you have to know the site of action and where the exact regulatory mechanism is, and the underlying secondary metabolites, whether it's cyclic AMP, ADP, whatever it is, you have to know the mechanism of action of insulin and glucagon. The next step is inhibitors. Say in glycolysis, there is enolase enzyme which is inhibited by NAF. So this is just one of the example. There are multiple examples. If you go to Harper, you can see at various steps a black bold link will be drawn and written negative acetate, so and so will be written. So you have to be thorough with all those things. Next is glycogen storage disorders. In any storage disorders or any other diseases you have to study in biochemistry, you have to stress on 2 points. One is the enzyme involved and the clinical features. So these 2 things you have to stress on. This is what I was talking about, glycolysis and gluconeogenesis. These are the irreversible steps. You have to be thorough with these 3 steps. And see, pyruvate kinase is part of glycolysis. Phosphoenol pyruvate carboxykinase is part of gluconeogenesis. So the enzymes involved, these 3 enzymes is part of gluconeogenesis. These 3 enzymes is part of glycolysis. You have to be thorough with it. Coming to fat metabolism. In this section, you have to, as I said, as any metabolic pathway, you have to first know the site. So whether intestine, it's liver or adipose tissue, you have to know what all reactions are occurring there. Second point, you have to know ATP production. In this case, calculation in beta oxidation, as I have already mentioned. The third point, regulatory steps. Hormone sensitive lipase, lipoprotein lipase, anything is, if at all any hormonal control is being exerted, you have to be thorough with that. Site I have already mentioned. Now in fat metabolism, you have to stress on lipoproteins and the hyperlipoproteinemia. You have to know the difference between VLDL, HDL, LDL, the apolipoproteins associated with them, and you have to know the disorders associated like familial hypercholesterolemia, Yeah, there is a table available and you have to be thorough with that table. Coming to heme synthesis, as I said, first you have to know the site. Then in ATP production is not important this, so leave that, not important. Next you have to know the regulatory steps, and in this case mostly instead of the regulatory step, what you have to stress is the disorders associated. Mostly two disorders are associated. First one is lead poisoning and porphyria. We will be visiting these two things shortly in the next slides. Coming to heme structure, you have to know you have to have a basic idea about the heme structure. Sometimes an image might come and you have to be thorough with it. So this is what I was talking about. See, sites. Now the steps. In this step, lead inhibit this one and this one. So you have to know these two enzymes. The rest, whatever it is, forget it. These two enzymes you have to know. And since this enzyme is inhibited by lead, this one accumulate ALA, and its level is measured in blood in case of lead poisoning. This you will be studying in general medicine. So, and the next topic you have to read is porphyria. It's coming over here. You have to know what are the types of porphyria and the enzyme involved. Two things, whenever a disorder comes, enzyme involved and the clinical feature. This is the same slide I've been talking about. Lead poisoning, two sites, and you have to be thorough with it. Coming to urea cycle, here also same site, any vital steps and disorders associated with it. And then another point, extra point you have to stress on is the source of nitrogen and the entry point. Here, these are the ways in which nitrogen is being expelled from our body, and these are the enzymes which are involved, and the vital steps which we have to focus is this one. OTC deficiency and CPS deficiency, the difference you have to know. Do you notice this one? This was AIMS May 2017 question. They were asking the role of phenylacetylglutamine. So in case of any urea cycle defect, you can provide this drug to bypass or to eliminate the excess nitrogen metabolites by urine. So this was asked in AIMS. So you have to know this one and this plus the sites where it's occurring. Coming to purine pyrimidine synthesis. First, you have to know the structure. How does adenine look like? How does guanine look like? How does cytosine look like? The difference between them you have to know. Okay, a mnemonic which you can remember is pure as gold. Purines, adenine and guanine, and it's small, so it has two chains. This is a basic, but now you have to know the difference between adenine and guanine, how to differentiate between them. You have to learn it. Now the next thing you have to stress on is the difference between purine and pyrimidine pathways. In this one I will be talking about it later. A slide is coming on. Okay, so let's discuss that topic there. Now there is also degradation pathway, and a disorder associated with is Lesch-Nyhan syndrome. So you have to know the enzyme and the clinical feature. This is what I was talking about. See this one? It's not seen in that one. Let's test. It's amine. Adenosine deaminase will lead to guanine. Now this was what I was telling. See in this one, ribose 5-phosphate. It's a first step in purine synthesis, whereas it's after completion of pyrimidine ring, that ribose 5-phosphate is added in case of pyrimidine synthesis. So you have to know the difference between purine and pyrimidine synthesis. Now coming to inborn errors of metabolism. These are the 4 disorders you have to be thorough with. Clinical feature, enzyme involved. Now coming to DNA. Now cycles are over. Now we come to DNA. It's a relatively simple topic. You just have to read it once. Nothing to memorize, nothing to by heart and all. Just basic concept in DNA is sufficient to crack AIIMS. These are the important topics which you have to stress on. One is the RNA types, and there is DNA polymerase, RNA polymerase types you have to know. There are other enzymes also involved, topoisomerase, gyrase. You have to know the function of these enzymes. Then you should have a basic idea about the difference between prokaryotes and eukaryotes, translation and transcription. Then this one is really important, epigenetic changes, slicing, methylation. You have to be thorough with the epigenetic modification that's possible and examples of it. Then coming to mutation, like addition, deletion, insertion, silent mutation, point mutation, lot of things are there, so you have to be thorough with this. And even an image based question might appear in this topic. Then there are multiple techniques, nucleic acid techniques you have to be thorough with. So in these steps you have to have a basic. First one, you have to see an image of this, so that if a spotter comes you have to identify. The second thing is you have to know how it's working, the mechanism. These 2 things you should be thorough with. So this was what I was talking about. An image based question can appear from it. What is this? What is that? Yeah. So just, it's very simple, but you have to see the image. Now coming to the third point, vitamins. In vitamins, 2 things you have to mainly know. One is the deficiency and the second one is the cofactor. Deficiency disorders, I think most of the guidebooks is providing it. It's very easy. You have to memorize a table, that's it. This cofactor, it's not mentioned well in most of the books. It's presented in a confusing manner, so I'll be talking about it in the coming slides. Another important topic is folic acid and vitamin B12, that is cyanocobalamin. You have to know the difference between them, how to identify it. So this one I talked about, this one I will be talking about, and the other one is collagen synthesis. You have to know steps of collagen synthesis in detail. Yeah, in detail you have to know the steps. So whenever you are studying a pathway, you have to stress on first is site, second is major steps. You have to know these 2 things in collagen synthesis. So this is what I am talking about. So in the next 3-4 slides I will be talking about the cofactors involved in multiple biochemical pathways. So for thiamine, it's easy to think in this manner: dehydrogenase, 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 3 dehydrogenase and 1 transketolase. That's for thiamine. Biotin mainly does carboxylation. So pyruvate carboxylase, acetyl coenzyme carboxylase, propionyl coenzyme carboxylase. So vitamin C. Vitamin C is hydroxylation. In collagen pathway you would have seen proline, lysine hydroxylation is done by vitamin C. There is another hydroxylase, dopamine beta hydroxylase, dopamine 2-beta norepinephrine. And another extra step which most commonly asked is iron absorption. Yes, vitamin C helps in iron absorption in the intestine. And vitamin K, there is gamma carboxylation of glutamic acid. It's important for the coagulation pathway. So one thing you would have noticed, like most of the biochemical reactions they have are almost similar by the words hydroxylation, carboxylation, dehydrogenase. You can guess which vitamin is involved in which one. So it's better to make a table out of it, which all biochemical reactions are helped or aided by these vitamins. So at the end, when you revise, it's so easy. Questions do come every year in AIMS exam. Questions do come from this topic. The next one is vitamin B5. It's the coenzyme A you are talking about. It's available in acyl transferase. So next is pyridoxine. It's mainly involved in transamination reactions. So AST, ALT levels, liver diseases, you would have heard about it. So the enzyme involved is pyridoxine, and it's also important in the synthesis of heme, norepinephrine, epinephrine, serotonin. Actually, the dopamine, norepinephrine, epinephrine pathway, you have to be thorough with this one. Like because like vitamin B6, vitamin C, a lot of things are getting involved in this pathway. So you have to know at exactly which step vitamin C is affecting, at which step vitamin B6 is affecting. You have to be thorough. The general topics. In this one, mainly you have to cover the enzyme kinetics, competitive, non-competitive, uncompetitive, and examples. That's one point you have to stress. The second type is the receptors, intracellular, extracellular receptors, and the various type, the secondary signal molecules, cyclic AMP, cyclic GMP, and examples of these receptors. You have to be thorough. In first aid, it's beautifully given. If you want, you can check it out. Then the next thing is the cell structure. You have to be thorough with this one. You don't have to go in detail, but you have to know like what's the function of ribosome, Golgi bodies. You have to be thorough with all these things. And another important topic which you will be coming across is lysosomal storage disorders. Just like glycogen storage disorders, you have to be thorough with clinical features and enzyme involved. Then another topic is flow cytometry. It's a practical point of view, it's important. You might come across a graph and asking to quote like what is the disease involved and all. So image you have to see, flow cytometry, and the concept, y-axis what it means, x-axis is what it means, you have to know. So this is the last slide, nothing extra. I think I have already mentioned it, like cycles, steps, and laboratory points. Image based question can come. Genetics, the mutations, I have shown you an image. Palindromic sequence, this is an important question. It was asked in AIMS May 2017. So genetic tRNA, you have to see the image. Karyotyping, it can be asked in path or biochem. You have to be thorough with it. Turners, downs, how do they look like in karyotyping? Vitamin deficiency disorders, an image based question may come. Maybe about riboflavin, showing the cheilitis and all. A question can come. Enzyme kinetic, the graphs, you have to be thorough with. Then the major genetic disorders like Downs, Patau, Edwards, you have to check out the image. Then next thing is the Marfan syndrome, rickets, zinc deficiency. Yeah, you have to be thorough with this zinc deficiency also. So whenever you come across the disorders, do Google image. If there is a characteristic image, save it. Have a collection of images. It will help you in revision. So I think that's all you have to know in biochemistry. Thank you.